Right, these are your chapter 14 notes. On genetics. Right, where did the PowerPoint go? I'm just patiently waiting. Okay, here we go. All right, you can read page 262 to 264 to understand the process of how Mendel came to understand some of our basic principles of heredity. I am going to tell you what those principles are, but if you want that history lesson of how he came to those ideas, these are the page numbers of your textbook. I know you're all itching to read that, but the idea here, for the most part, was that he worked with pea plants. And this was really easy to work with as an organism for him because they reproduce very quickly. They have very limited phenotypic traits, and most of those traits are either dominant or recessive. There's not a lot of other things that we now know, like incomplete dominance or codominance or epistasis or anything like that. So they were really easy to study. He could really easy control the mating of them, unlike like rabbits or mice, a little bit harder. But he could physically move the pollen of one flower to the stamen of another flower to mate them. And what he ended up with is these are some of the traits he had. They had smoother, wrinkled seeds. Their flowers were either axial or terminal in their positions. They either were white or purple flowers. Their seed pods were either yellow or green. And so there were some very limited traits, but they were very clear. They were one thing or the other. There was not 3 or 4 different traits. It's kind of like eye color. We still don't really understand that because there are so many versions of it. So he studied pea plants with very simple traits. He came up with the term true breeding. Over many generations of self-pollination, the offspring always looked like the parent. Hybridization, mating or crossing of 2 true breeding plants. You might hear these terms also when you talk about like dog breeding, because they do a lot of true breeding. For example, the line of the Cocker Spaniel has always been Cocker Spaniel, and they've never mixed with anything else. That would be a true bred Cocker Spaniel. I'm sorry. Hard for me to do this one. Some terms you need to understand, especially when we get to fruit flies. The P generation is when you are mating 2 true breeding parents. So that means that they've never been mixed with anything else. They're definitely one trait. And then the F1 generation is going to be a hybrid of the offspring of the P generation. So when you make the P generation, you're going to get the F1 generation. And then when you make the offspring of the F1 generation, you're going to get the F2 generation by doing that. And we'll do that with fruit flies. So what he had in his P generation, he had a true breeding purple and a true breeding white. And funny, all of them turned out purple. He's like, well, why aren't there any white ones? But when he bred the F1, so he took 2 F1s and he bred them together, all of a sudden he saw this ratio of 3 to 1, 3 purple to 1 white. And he said, wow, why is that? Why is the white seemingly skipping generations? Well, we now know that when we see that skipping of generations, the trait is recessive. But he didn't really understand that then. He just was trying to figure out why it did that. So you notice that 3 to 1 ratio is consistently in the F2. He kind of came up with these ideas. So we now have put vocabulary behind it. Alternative versions of a gene account for the trait variations in inherited characteristics. These alternative versions are now called alleles. He just knew that there was something. There were alternative versions. There wasn't just one. There was purple and white. For each character, an organism inherits 2 copies of a gene, one from each parent. He came up with that idea too, and we know that now to be consistent with what he observed. If 2 alleles exist two alleles exist at a locus are different than one, which we now call the dominant one, will determine what the organism's appearance is. The other has no noticeable effect on the appearance. We now know that one to be a recessive. So those are the vocab terms behind what he came up with. This is something we now know too. The law of segregation, the two alleles for heritable characteristics segregate or separate from each other during gamete formation, which this term is called what? Meiosis, I hope you said that. And end up in different gametes, either the egg or the sperm. Only gets one of the two alleles present in the somatic cells. So this is what he's talking about here. So these are alleles. And remember that the reason that there's two copies here is because in interphase, copy our DNA. Okay, so this big G is a copy of this one, and this little G is a copy of this one. So when these separate out, you're going to get the big G separated out and then the little G separated out. That's the law of segregation. They're going to separate out. And the way we show that in a Punnett square is on one side of the Punnett square, we put these two options. And then on this side, or sorry, my apologies. I'll erase that if I can. On this side, we put this two. And on this side, we put these two. And then we see where that combination will lead us in the offspring. And you'll see that when they combine, you're going to get a hybrid. So a Punnett square is a device that we use for predicting what the offspring of a cross are going to look like, or in what their genetic makeup is going to be, if we know the genetic makeup of parents. This is a Punnett square. You've seen them before. This is probably nothing new this year. However, we're going to do a few of them physically drawing out these boxes, but then I'm going to teach you a faster way, because when you get talking about 3 or 4 or 5 traits, these boxes become huge and they're just time consuming. And there is a faster way to do this. So let's get a couple of pieces of vocabulary down. If you don't know this vocabulary, it's going to be very hard to determine what these alleles are here. If I tell you heterozygous and you don't know that means one capital and one lowercase letter, you're going to be lost. You can't do the Punnett square because you don't even know how to set up the Punnett square. So make sure you feel these terms in your brain. Homozygous, an organism that has a pair of identical alleles for a character. So that what we're talking about here is some kind of trait we're looking at. So 2 big R's or 2 little r's. This means that they're dominant, and this means that they're recessive. I always remember this because of this term. Remember, this is meaning same or alike. And then heterozygous, an organism that has 2 different alleles for a gene. So big R and little r has both. Now, this is the dominant and this is the recessive. You are going to see the dominant form because you always see the dominant when both are present. The recessive form is only going to be visible if you have 2 recessives in the organism. And this I always remember because those hetero mean different. Phenotype means the appearance. So what are you actually going to see, red or white? These are going to be the words, okay? And then the genotype is going to be the actual physical letters of what they look, what their genetic makeup is, what genes or what alleles they have. Make sure you know these vocabularies, and you can use them. Pretend we have a mystery plant with purple flowers. These particular flowers come in either white or purple. And purple, we know, is dominant. To determine the genotype, we can cross the purple flower with a white flower. We call this a test cross to figure out what the genetic makeup of this mystery plant is. Excuse me. So a test cross is always crossing your unknown with a recessive. Okay, a homozygous recessive to be exact. What would we expect the outcome to be if the mystery plant is big P, or big P, little p? So kind of mentally do that. I'll give you a minute. Okay, so big P, right? And we're crossing it with a little p. We would expect all of the offspring to be purple, right? So when we do this cross, if all of our offspring are purple, then most likely our mystery plant is big P. However, if we cross this, we would get about half of them being purple and the other half of them being white. So if we have about a 1-to-1 ratio, or about 50-50 white and purple, then we know that our mystery plant is heterozygous. That's how our test cross works. You cross your unknown with a homozygous recessive to figure out if your unknown is homozygous dominant or heterozygous. A test cross is when a geneticist breeds an organism of known phenotype with a recessive homozygote because it can reveal the genotype of that organism. That's just what I said. And the easy answer. A monohybrid cross is a cross between two genotypes following only one trait. A dihybrid cross is a cross following two different traits. So here's a monohybrid. We did a lot of these in Bio 1. We put the possibilities here for the gametes of, it doesn't really matter. We can put female up here and male to the side, or male up here and female to the side. It doesn't matter. And then the possibilities here. Okay. Where do those come from? It comes from their genotype. Okay. Go back just a minute. It comes from right here. You have the possibility of this or this. Okay. And this could be either egg or sperm. It comes from meiosis. What are the possibilities? So that's where you get those. This is a monohybrid cross. It's very easy. You combine the letters in each of the boxes, and then you should know the phenotype based on what you know about what's dominant and what's recessive. A dihybrid cross, which we're going to get into these this year, is when you're using two different traits. So here, we're looking at the color, which it looks like it's either yellow or green. It looks like green is recessive here. And then we're looking at round or wrinkled. And it looks like, usually if you use the letter of the dominant form, so it looks like round is dominant. And so in this diagram here, we are combining round and wrinkled in these boxes. And then we're combining those boxes, and so now we have a round yellow, round yellow, round yellow, round yellow. Down here we have wrinkled in green and wrinkled in yellow. So now you can get a a ratio that's a little bit different. The law of independent assortment states that each pair of alleles segregates independently of each other during gamete formation. So again, gamete formation is what? It's meiosis. I hope that's what you said. Okay. So this is going back to what you learned about meiosis. In other words, when crossing these and crossing these and these are separate events. Okay. This Y doesn't always go with this R. Okay. It could also go with the recessive R. And the recessive Y doesn't always go with the dominant R. It could go with the recessive R. And so it's kind of like algebra when you're doing these dihybrid crosses. It's like, I think you guys call it foiling, where you actually multiply this by both, and then you multiply this one by both. Well, it's kind of the same idea when you're setting up these Punnett squares. It only applies to genes located on different chromosomes. This becomes really important because when we talk about genes that are on the same chromosome, they usually do go together in a gamete because they're on the same chromosome. This applies to different chromosomes, and we're going to talk about this much later. It's called linked genes. So here's independent assortment and kind of showing you the idea of what that means. When these line up in the middle, notice that on this side you got the 2 orange and the 2 blue, and this side you got an orange and a blue, and a blue and an orange. So what that means is here, if you're looking at straight, you might have brown eyes and black hair, where here you have blue eyes and red hair. But if these are sorted differently, you could end up with brown eyes and red hair, or blue eyes and black hair. Well, this is only 2 pairs. Think about how many different arrangements you can get in the gametes when you have 23 pairs. It gets pretty hairy now. And there's actually an algebraic, and I guess you could call it algebraic. There's a mathematical expression for knowing that answer. Let me know in your notes if you know the answer to that, how to get that. Okay, so let's do a little bit of monohybrid practice problems from Bio 1. It should be pretty easy. We have flower position axial and terminal. Notice that axial has a capital letter, so that means it's dominant, and terminal has a lowercase letter, which means it's recessive. So go ahead and see if you can do those problems. So I'll do one of them here. And I'm going to combine these 2 boxes and these 2 boxes and these 2 boxes and these 2 boxes. So what I get here is I get, and there's a lot of ways you can express this. I'm going to express it as fractions. You get half being homozygous dominant and half being heterozygous. But what that means is, because they both have the dominant, that 4 out of 4, or 100%, are going to be axial. All my flowers are going to be axial. Doing this one here, this now gives me 1/4 of one of the 4 boxes being A, 2 of the 4 boxes being little a, and we can actually simplify that to 1/2. And then 1 of the 4 boxes being little a, little a. So this means that 3/4 are going to be axial, so about 75%, and 1/4 are going to be terminal, which is about 25%. And you can express that in different fractions as a percent, like I did verbally, or you can do it as a ratio. For example, this is a 1:1 ratio, right? In the genotype, and in the phenotype, it is a 3:1 ratio. Go ahead and see if you can do these. Here you should have gotten all are going to be round and heterozygous. Here you should have gotten similar to here, that 3:1 ratio. 3 of them are going to be round and 1 of them is going to be wrinkled. Round. And you've got 1/4, 1/2, and 1/4. Okay, now let's try a different one. This one's a little bit more challenging because it requires you to do a dihybrid cross. So when you're setting up a dihybrid cross, you're going to have more boxes. Let me see if I've got. Well, let's see. I'm trying to decide where I want to go from here. You do need to know how to set up a dihybrid cross, although it's the long way of doing it. To set up a dihybrid cross, you're going to multiply, essentially, the big A with both of the two R's, and then you're going to take the little a and you're going to FOIL that. So you're going to end up with big A, big R, big A, big R, little a, big R, little a, big R. And then over on this side, you're going to end up with big A, big R, big A, little r, little a, big R, and little a, little r. So then you would set up a dihybrid cross. I don't have a whole lot of room here. A cross with this information, and you would fill them in. So you'd have big A, big A, big R, big R, big A, big A, big R, big R, big A, little a, big R, big R, big A, little a, big R, big R, big A, big A, big R, little r, big A, big A, big R, little r, big A, little a, big R, little r, big A, big A, oh wait, that's big A, oh no, big A, little a, that's little a, R, R. And then you could go ahead and completely fill this in, and I want you to do that. Okay, and then go ahead and try to figure out what your phenotypic ratio is. Remember, phenotypic ratio means what do they look like? How many of this? How many are going to be aerial and round? How many are going to be axial and round? How many are going to be terminal and round? How many are going to be axial and wrinkled? How many are going to be terminal and wrinkled? So that takes a lot of time. It's very time consuming to do Punnett squares. Okay. If we memorize some of the simple monohybrid crosses, which we're going to do, and you're going to take a quiz on it, so that I know you know them, it's going to make it really easy to just do this multiplication-wise. So memorizing these monohybrid crosses, and it doesn't matter what letter you use, I'm just using R here. Anytime you multiply two homozygous dominant, you are always going to get 100% two homozygous dominant. If you multiply a homozygous dominant by a heterozygous, you're going to get half of them being homozygous dominant and half of them being heterozygous. If you multiply homozygous dominant with homozygous recessive, all of them are going to be heterozygous. If you multiply two heterozygous, you're going to get 1/4 homozygous dominant, 1/2 heterozygous, and 1/4 homozygous recessive. This is going to be the most variety. You're going to get the most options and offspring by doing two heterozygous. So by a heterozygous, by a homozygous recessive, you're going to get half heterozygous, half homozygous recessive. If you multiply two recessives, you're going to get all recessive. You need to memorize these. We're going to take a quiz. Okay, and you're going to have about 5 minutes to write them down. So you need to know them. When we know those, we can use something called the multiplication rule to multiply the probability of one event by the probability of another event. It's kind of like the coin landing on heads up twice. There's a probability of it landing heads up 50% of the time in one flip and 50% of the time in the next flip. So together, for it happening twice, there's 1/4 chance that you're going to flip that coin twice, and both times it's going to land heads up. So it's the same idea here. The probability of getting a heterozygous female egg, the probability of getting the recessive allele is 1/2, and the probability of getting the male sperm getting the little r is also 1/2. So the probability of them coming together when you're mating two heterozygotes is 1/4. It's the same thing as doing a Punnett square, except you don't actually have to physically write down a Punnett square. There's also the probability, or sorry, the addition rule in the laws of probability, and that means that that's the probability that any one of two or more mutually exclusive they're not occurring together, events will occur is calculated by adding their individual probabilities. So to determine the probability that the F2 generation will be heterozygous, if the parents are both heterozygous, then we know that the offspring, big R, little r, can get the big R from either mom or dad, but not both, because they don't both have that. And the same applies to little r. So the probability is 1/4 that you're going to get big R, little r, and 1/4 big R, little r. Add those together, and you will get a half possibility of getting big R, little r, which is what you would get if you did a Punnett square for this. This is just showing, it's kind of relating the idea of segregation of alleles to coin tosses. Okay, it's like heads or tails. You're going to get a half each time, because remember, there are going to be, in every instance of meiosis, there are going to be 4 sperms, and 2 of them are going to look like one, and 2 of them are going to look like the other, because they're identical copies of each other, because of that replication thing that happens in interphase. So let's do a couple of examples where we don't set up a Punnett square, we just try to do the multiplication. So here we have round and wrinkled, in green and yellow. So this is the shape, and this is the color. And we're going to make a homozygous round with a yellow plant. So homozygous round, you can guess that is going to be like this. And it's yellow, so that means that in this case it's recessive. So it's going to be like this. Okay. And then, I'll write that in here. And then we're going to make with that a wrinkled homozygous green. So wrinkled means it's going to be like this, and homozygous green means it's going to be like this. Okay? So we're going to go ahead and we're going to multiply these out. And instead of doing the whole Punnett square, we're just going to do the math. And we're going to give the phenotype and genotype specific ratios for this. And then I'll let you get on with the next one here. So we know that within this, there is 1/2 chance of getting big R, little r, and 1/2 chance of getting little r, because we've memorized that. So we know that. And then here, we know that the only possibility is big G, little g. Okay? So remember that here we're going to do the multiplication. What offspring are going to look like. Hang on 1 second, sorry. Okay, sorry, I was interrupted there. So where was I at? Okay. Let's see. I lost that. All right, so we're just multiplying these together because of that rule of multiplication. So what's the chance that we're going to get big R, big G, little g, and what's the probability we're going to get little r, big G, little g? Because those are the only combinations given this cross. So this probability multiplied by this probability, and this probability multiplied by this probability, it means that we're going to get a 1/2 chance of getting this and a 1/2 chance of getting this. So sometimes you're going to be asked how many offspring. So say that we have 100 plants that are produced from this farm. That means that 50 of them are going to be this, and 50 of them are going to be this. So you have to be able to take that percent or that fraction and turn it into a physical number if you're given the number of offspring. Here's another example, tall and short. And we're actually using tall, round, and green here. And then we're using short, wrinkled, and yellow. So we're actually combining 3 traits now. What are the chances of getting this offspring? So we could actually sit here and multiply it out to figure out every single possible offspring, but they're just asking us about one. So that makes it a whole lot easier. So from this cross here, what's the probability of getting this? Well, that's a 1, right? 4 out of 4, because that's the only thing you can get. So we're just going to do a 1 here. And then big G, little g, what's the probability of getting that? Well, remember from memorizing, that's a half chance you're going to get big G, little g, and a half chance you're going to get little g, little g. So we'll call that a half. And then little r, and we've got this and this. And we know from memorizing that there's a 4th chance of getting little r, little r. So you're going to multiply all of these together. And there's a 1/8 chance that you're going to get this offspring from that cross. That goes so much faster than doing an entire Punnett square or a dihybrid, trihybrid, in this case, cross, and trying to find and count up how many boxes there are. Okay, so what I'm going to do is I'm going to stop today, and then we're going to continue on with complex inheritance beyond just this stuff, this dominant and recessive, in the next